Hello everyone, so this is going to be the second to last chapter for exam 2 from the professor's notes for biochem 2. Going to run through this quickly. I was getting caught up doing something else, so yeah, let's just run through it. Alright, so we're going to use amino acid oxidation when the degraded peptide ones aren't needed, we have an excess of dietary amino acids, or the cell proteins are used in absence of carbs. So that's the only time when we're going to be breaking down the amino acids is when we don't have carbs or we don't have lipids, because it goes carbs to lipids to proteins. And you're breaking down the proteins here. You're in a lot of trouble there because you're literally going to start breaking down the cellular material as an energy source. So like you're eating your own self, basically. Don't want everyone to be in that situation, and hopefully you're never in that situation too. Alright, so plants are going to conserve all nitrogen. Aquatic vertebrates are going to be releasing ammonia in the form of NH4 plus, not NH3, NH4, because this is going to be at the physiological pH. They're going to be throwing that out into their environment. Passively, you're going to be seeing this exchange in the epithelial cells, and then actively it will be through the gills for aquatic animals. Land animals and sharks are going to be secreting urea. This is less toxic than our NH4, and it's also soluble. Reptiles are going to make this uric acid paste, and that's going to be a little bit insoluble. And they do this, so reptiles is also talking about birds, because birds are also reptiles. This is going to be the usage of uric acid because they want to conserve water. This is their way of doing that. Okay, humans, though, and our great apes, our relatives, the great apes, so humans and the great apes, we're going to have both urea from amino acid waste and uric acid from the purines. So we're going to be doing both uric and urea. Alright, the first step in the oxidation of amino acids would be to remove the amino group because NH4 is so toxic. So you're going to be having this amino acid, and you're also going to have alpha-ketoglutarate present somewhere. And the enzyme involved is an aminotransferase. Aminotransferase, what will happen is it's going to have this PLP, this pyridoxal phosphate cofactor. All aminotransferases have this. They're also known as transaminases. So all of these types of enzymes, aminotransferases, will have this PLP cofactor, this pyridoxal phosphate. And so basically you're just going to transfer the amino group from the amino acid to the alpha-ketoglutarate. When you do this, your product will be L-glutamate. So you're going to make glutamate. The second transfer of an amino group to the alpha-ketoglutarate, so after we get to our glutamate from this first transfer, if we do a second transfer, we're going to get to glutamine, and we'll get to that reaction in a bit. More on pyridoxal phosphate. PLP is also known as vitamin B6. It again transfers the amino group, or the NH2, to alpha-ketoglutarate as an aldehyde. In the result of this, we also have this thing called an alpha-keto acid. That's just an amino acid without the amine group on it, so it's called an alpha-keto acid. Alright, so let's go and talk about the reverse of step 1 here. So let's say we had glutamate, and we want to get to our alpha-ketoglutarate. So what we would use is this glutamate dehydrogenase. And when we use glutamate and this dehydrogenase, we get our alpha-ketoglutarate as well as NH4. This NH4 is going to go to the urea cycle. And the reverse reaction of this, of course, will happen in the mitochondrial matrix. NAD is needed for it. And we got some, considerations to take on. So, with high ADP, this glutamate dehydrogenase is on, you're going to make alpha-ketoglutarate. With high GTP, this will equal off because this means we already have a lot of alpha-ketoglutarate. So therefore, this glutamate dehydrogenase will be off when we have a lot of GTP. Alright, step 2 would be taking our glutamate, using this glutamine synthase. Actually, glutamine synthetase, sorry about that, it's a synthetase, so we need some ATP. And then when we use this synthetase of glutamine on glutamate, we're going to be able to form glutamine. The reverse step would be taking the glutamine, and then we're going to use this enzyme called glutaminase. And that's going to form glutamate and NH4. The reverse of glutamine to glutamate, that reaction, so glutamine treated by glutamine treated by glutaminase will result into glutamate. That reaction takes place in the liver and kidney. Alright, let's switch to a different amino acid. Let's talk about alanine. So alanine transports NH4 from the muscle to the liver. The muscles will make pyruvate lactate and our ammonia, or NH4. And we have this reaction here. So alanine, when treated with alanine aminotransferase, do you see that? Aminotransferase. That means we'll need PLP in this reaction. When we treat aniline with its aminotransferase, we will get our pyruvate. And of course that pyruvate could go through the cycles that deal with carbs. At the same time, whenever we have this aminotransferase, any type of aminotransferase will also have this reaction too. We'll have this alpha-ketoglutarate becoming glutamate, because again, there's the aminotransferase involved. And so those amine groups are going to be shuffled around, basically. They're going to be playing ping pong, as my friend Umar said. So NH4 will be excreted. Pyruvate again go to form glucose and the pathways that involve it. Let's talk about NH4's toxicity. So why it's toxic. It competes with K+, the ion, in astrocytes, so it causes this cytotoxicity with the neurotransmitters, so that's a very dangerous thing to watch out for. Okay, I don't know if I want to cover the urea cycle. Oh yeah, let's do the urea cycle. So urea cycle, we're going to be dealing with that NH4 again. The NH4 of hepatocytes will go into urea after this urea cycle, and then that urea will go into the bloodstream and it's going to be excreted out. So we have this one transition step in the mitochondria, so we could get it out into the cytosol by step 2 of the actual urea cycle. So the mitochondria transition step is taking our NH4 plus a bicarbonate, HCO3, plus 2 ATP. We need 2 ATP for this process. The enzyme that we're going to be using is carbonyl phosphate synthetase 1, and when we use this enzyme, we're going to make carbonyl phosphate. Alright, so that's our transition step. Okay, step 1 of the actual urea cycle is our carbonyl phosphate plus ornithine. This is a new molecule. I've never heard about it. So let's see. We're going to now use ornithine transcarbamylase. I probably butchered that, but we're going to use ornithine transcarbamylase. We're going to make citrulline. Okay, so step 2. Now in the cytosol, citrulline will be treated with aspartate. Oh wait, what is this? Citrulline will be treated with arginine synthetase, so we need ATP, and we also need aspartate. That's what I was trying to say there. Sorry about that. And this is going to form this molecule here called argininosuccinate, and this is a condensation reaction. Okay, step 3, we have our argininosuccinate. I think that's how you pronounce it. We're going to be using an argininosuccinate-ase as the enzyme, and we're going to be forming arginine and fumarate. So fumarate, like the fumarate from the what's it now? Shoot, why is it blanking on me? Fumarate from the citric acid cycle. There we go. Sorry about that. Okay, number 4, we're going to be dealing with just the arginine that we've made from the arginous succinate ACE. Alright, so we're going to take our arginine, and we're going to use an arginase as the enzyme, and we're going to get urea, as well as ornithine. So remember, ornithine was in this first step here of the urea cycle. We had to combine ornithine with carbomoyl phosphate, and then the ornithine transcarbamylase will make citrulline. And then the cycle just happens. That's why I have another cycle after you get to step 4 of the arginine and the arginase, and then you get urea and ornithine. The ornithine cycles back to step 1. Okay, let's link the citric acid cycle and the urea cycle very quickly here. So citrulline to arginine succinate, that was step 2. But also we have this process here, oxaloacetate with a transaminase will make aspartate. Aspartate will then go to step 2, because remember step 2 required aspartate, so that's how we get our aspartate. We have oxaloacetate and a transaminase, and then it will form aspartate. We also have this process here, the arginine succinate. Then we go to fumarate. That was step 3. The enzyme required was arginous succinate ACE. And from the fumarate, we go to malate. Like in the citric acid cycle, the malate will be going into the mitochondria, and then that becomes oxaloacetate. And then again, it's just all connected. This interconnected part is called the malate aspartate shuttle, which is pretty interesting. There's like a linkage there between the two. Okay, let's talk about regulation of the urea cycle. So you regulate the 4 enzymes of the cycle itself, as well as the carbamoyl phosphate synthetase 1. How do you regulate this? So you have this molecule called N-acetylglutamate. This will activate the carbamoyl phosphate synthase. Is it a synthase or synthetase? Let me go back. Sorry about that. It is... Where am I? It is a... Where did I just saw it. Did I like erase it by accident? I don't think so. It's a synthetase, so the carbamoyl phosphate synthetase. It's a synthetase, so we need ATP. Alright, so N-acetylglutamate will activate the CPS1. That's, again, the transition step before you get into the urea cycle. How you form N-acetylglutamate is this process. Alpha-ketoglutarate plus glutamate with that N-acetylglutamate synthase will make our N-acetylglutamate. N-acetylglutamate says that the urea cycle is needed for that instance, so that's how you regulate the urea cycle. Alright, thanks so much for tuning in. Please do something nice for someone and take care.